Okay, good afternoon, class. Nice to meet you again for the discussion. So we will continue with question number 4. Question number 4, quite a lot of things to learn. Then let's read together. Question number 4. Okay, so let's read the passage first. A subspecies is genetically distinct population within a species that is, there's some phenotypic differences from the rest of the species, but it's not yet reproductively isolated. So this is talking about the subspecies that we have learned in the syllabus. So let me show you since we are touching it. Okay. So this is the vertical line downwards is showing with regards to time how from a homogeneous ancestral population, a single population of same species can slowly become different species, so species A and B. So it has to go through a long period of time where first they are separated geographically, so they become two different populations, and then subjected to the local selection pressure, which is different on both sides. So then there'll be less gene flow between the two populations, so they become two different races. So the less gene flow, it can be because they're separated by some isolating mechanism, especially geographical isolating mechanism. So here, geographical isolating mechanism established. So less gene flow means there less migration of individuals between these two races, and there's less interbreeding between these two races. So that's the cause of gene flow, migration and reproduction. Okay. So then after a long period of time, then pre-zygotic isolating mechanism becomes established. So if you want to know again what is pre-zygotic isolating mechanism, let's go back to here. So these are all the pre-zygotic isolating mechanism. That means the two individuals are prevented from being interbreeding, either because of geographical, ecological, means in the same forest, in the same place, they have different role. They live in different location, like on top of the tree or the bottom of the tree or underground. Okay, so different niches. Okay, ecology means different niches. Then temporal, it means mating season is different. Behavioral mating call is different. Morphological means the copulation organ is different. Gamete motility means the gamete sperm that goes into the female reproductive organ cannot survive, so they die. Okay, so all these will cause the two races to become two different races, two different subspecies. Sorry, it's actually subspecies. Where's that thing here? Okay. So two different subspecies arises because of pre-zygotic isolating mechanism. Then they continue to be subjected to selection pressure at the local environment, and eventually post-zygotic isolating mechanisms established to become two different species, totally no gene flow. Okay, so showing the processes from same population to two different population to two different races, two different subspecies, and finally two different species with decreasing gene flow. Okay, so that's about the theory of the question. So let's look at this. So they said there are 9 subspecies of the tiger, Panthera tigris. So this is a species name. And it have been identified. 6 of these subspecies are found on the mainland Asia, and 3 of the subspecies originate from Sunda Islands. So out of the 9 subspecies of this tiger, Panthera tigris subspecies, out of the 9 subspecies, 3 of them are found in this Sunda Island, which is made of these 3 main islands here: Sumatra, Java, and Bali. So therefore, over here, they mentioned that all these 3 islands, they have their own subspecies of the Panthera tigris. So they actually named the subspecies with an additional name, if you can see here. Okay, so Panthera tigris is the species name, and then the last one, Balaka, because it's in Bali. PT Sodaika, because it's in the Javan, and then PT Sumatrae, because it's in the Sumatra. So these are the subspecies name of the species here. Okay, so let's look at the question next. Okay, part 1, suggest and explain how the 3 subspecies of tiger on the Sunda Islands are formed. That means from original same species, how these three subspecies in three different islands are formed. So this one, you go back to the theory, it has to do with the allopatric speciation. Okay, allopatric speciation, which is shown here. Okay. So the keyword is, number one, you must talk about there is a geographical isolation, right? This original population, original species, they start to move to different locations, and these three islands were once connected together. So they move away to three different locations, and then eventually when the three locations are separated by sea or ocean, it leads to geographical isolation. Okay, so the sea that eventually separate the three location results in geographical isolation. So once the geographical isolation, there is no gene flow. That means the two populations, they will not migrate, they will not interbreed. So there will be no gene flow between the three populations in the three different islands. Let me put back here. Okay. Okay, here. So they're separated to three different locations, and then the sea is formed. So it leads to geographical isolation. And then there's no interbreeding among them, so there's no gene flow. Okay, second marking point. Then in the local environment, Sumatra, Java, Bali, they have different selection pressure. So geographical isolation, and then no gene flow between the islands, the three of them, geographical due to the ocean. So your answer must be very crystal clear to the point. Geographical isolation, then you must say what kind of geographical isolation. So here is ocean. Sometimes it can be mountain, sometimes it can be desert, right? Or lake. So you need to mention clearly to make sure you score the marks, okay? Because the answer said refer to. Refer to means if you write geographical isolation, you will not get the mark. You write geographical isolation due to ocean, yes, you will get the mark. Okay, it means you're referring to it is the ocean that caused geographical isolation. No gene flow, then you must say no gene flow where? Between the islands. So must be very crystal clear. So in different island, the different island has got different selection pressure. Okay, different selection pressure, which is actually the environmental factor. So you can also say environmental factor. Okay. So after that, next, there is a possibility, it's all possibility that can cause them to become different mutation. So two things can cause them to be genetically variable. One is because of different selection. Pressure will select different allele. Mutation create new allele. Okay, creates new allele. Different selection pressure will select different favorable allele in different island. Okay, in different island. So this is okay. So individual with favorable allele will survive. So you know that this you need to mention survive and reproduce. Okay, and then therefore they will pass on the favorable alleles to the offsprings. So these are the standard point you need to mention in the evolution chapter. Okay. So up to this stage, you need to find complete the answer by saying why they become different species. So they become different species because reproductively isolated. So of course we know it's due to pre-zygotic, which include pre-zygotic and post-zygotic isolation. So isolation means isolation isolation from interbreeding. So this is called reproductive isolation. Okay, so therefore, hence different species. So this is answering the question, right? Starting from they are the same species, you explain step by step until they become different species. Of course, this entire process here has got a name to it, so it's called allopatric speciation. So this is 1 mark. Okay, so very easy, yeah. Okay, next question. Part 2. Okay, explain why specific primers were used for tiger mtDNA sections. So you need to read the passages, right? Okay, here. So in this passage, it says that recently there was a study being made, okay, to analyze the 9 subspecies of the tiger. So they look at specific section of mitochondrial DNA. Remember, what are the methods that we use to find out genetic relatedness between individuals? And from the genetic relatedness, we can find out the evolutionary, we can draw the evolutionary tree and find out the time of divergence in evolution, or when did they have common ancestor. Okay. So the method to find out genetic relatedness by finding out the similarity in their mitochondrial DNA. And from there, we can draw the evolution tree by finding out the time of divergence in evolution, means when did they have the common ancestor. So the more closely related they are, the more recent is the ancestor, right? So more closely related, more recent is the common ancestor. So this is the principle of the test. So now they take mitochondrial DNA, okay? So they take the mitochondrial DNA from the tiger sample and they amplify the DNA with PCR method, and then they compare the mitochondrial DNA to assess the evolutionary history. So they want to find out the evolutionary tree. Yeah? So the source of DNA for the extinct subspecies came from the museum, okay? And then mitochondrial DNA was extracted and they used PCR to amplify it. It means to make many copies of it, okay? And then they use primer. They use primer based on specific section of tiger mtDNA. So you know that primer is a short single-stranded DNA with complementary bases in order to bind to a specific region of the mitochondrial DNA. So mitochondrial DNA is a circular DNA, so let's draw circular DNA, okay? And it is also double-stranded, so let me draw. I hope I can get it drawn double-stranded. Oh no. Okay, like this, double-stranded. Okay, so the primer is supposed to bind to a certain part of the mitochondrial DNA. So let me draw the primer in blue color. Blue color. Okay, so let's say I put a primer here. It's supposed to bind to this region because it's got complementary bases with this region. Okay. Okay, that's all I can draw. Complementary bases. Okay. So therefore, that means for all the DNA they've extracted from all the 3 different subspecies of tiger, right? We take the mitochondrial DNA and we're going to stick the primer to a certain region, and then we're going to amplify it. So because you know that when we do PCR, right? During PCR, we need to use the primer to bind to specific region of the DNA before elongation occurs. Okay? So that means so that we want to examine how similar it is in terms of the base sequence in this section of mitochondrial DNA among the three different types of subspecies. Okay, so they obtain it from the three island subspecies, uh, and they found that this mitochondrial DNA section, this section here, they are very similar, only this section. So let's look at the question. Explain why specific primers were used for the tiger mtDNA sections. 2 marks. So let's look at the mark scheme. Okay. Why we use specific primers? Because the primer is able to bind to the complementary base sequence in mitochondrial DNA. So that once it binds to there, that means we will only want to amplify that section of mitochondrial DNA. So we only want to amplify the specific mitochondrial section that is mentioned in the passage earlier on. Okay? To amplify the specific, where is it? Specific section. Okay? Specific section. So we want to amplify that section. For what purpose? Because this is used to find out the genetic relatedness, right? So to find out to compare this section among the three species in terms of their similarity and differences so that we can find out how closely related they are among the three subspecies and find out their common ancestor, how close in terms of time in their common ancestor. Okay, so you can see that when you write the answer, you need to relate the answer to the scenario of the question that they say, that they do this PCR method in order to find out how closely related they are. You look at similarity and differences between the base sequences of the DNA. So to that specific detail of understanding, you need to know how I know they are related by looking at similarity differences in the base sequence of the mitochondrial DNA section only, not the entire one. In this particular example, okay? So next question, A part 3. Describe and explain one characteristic of mitochondrial DNA that makes it more useful than using nuclear DNA to provide evidence of this evolution. Now over here, some students did not write their answer based on the question. You see, the question say, describe and explain one characteristics only. And that is why 2 marks is given. So some students didn't get 2 marks because the student described 1, 2, 3, 4 characteristic but did not explain. So when you describe the characteristic, you did not explain, you cannot get the full marks because the question is describe and explain one. So let's say you describe A and then you explain B, you only get 1 mark. Okay, because you must explain the same thing. So that is where you need to pay attention to answering questions so that you answer the question to score the marks and you don't write unnecessary things, okay? So why do we use mitochondrial DNA? Because in one cell, there can be more than, in one cell, there's only one nucleus, right? But there can be many mitochondrion. So therefore, there are more copies of the mitochondrial DNA that we can isolate from the cell than the DNA from the nucleus. So we have large quantity of the mitochondrial DNA in the cell. Okay? So when you got large quantity, it is easier to extract because you've got many mitochondria. So it's easier to extract, easier to amplify for DNA testing. Okay, so this is one set of answer. Second set of answer is mitochondrial DNA is smaller than all the DNA in the nucleus. Okay, it's circular DNA, so it's got small genome size. Small genome size means a small DNA. Okay, genome is all the DNA, all the genetic material, in this case in the mitochondria. So it's smaller. So it is easier for us to do what? When it's smaller, it's easier for us to locate the specific section of DNA to test because we only want to bind to specific section. So this is one set of answer. Next set, mitochondria DNA is a single copy of DNA. There's no a pair. Like chromosome is always a pair. In the nucleus, if you take the DNA from nucleus, it's always a pair. We have got maternal and paternal chromosome as a pair, right? So we've got maternal chromosome, okay, maternal chromosome, and then we have paternal chromosome. It's always a pair. Whereas in the mitochondria, we only have got a circular DNA and nothing else. Circular DNA. The main chromosome is circular DNA, ignoring the plasmid. Plasmid can be a few copies. Main chromosome is only one. So it is a single copy of the DNA here, meaning that there is no paired alleles. It exists in a haploid state. So what is the advantage of this one? So this is the feature. So feature explanation. So that means there will be no change in the alleles from generation to generation. Because in sexually producing organism, the gametes can be genetically variable because of meiosis. But over here, there's no exchange of alleles because of crossing over, independent assortment and all these things. So the genetic material remain the same, quite stable throughout generation, unless mutation occurs. So we want that stability there so that we can trace the genetic relatedness. So from the same family, maternal family, all the lineage from the maternal family, all of them will have no change in the in the base sequence of the mitochondrial DNA due to passing on of the DNA to the offsprings by binary fission. Okay, binary fission. So there's no change. So this is comparing with what? Comparing with sexually producing what? There's a change. So binary, we can also say binary fission. So binary fission does not involve what? No meiosis. No meiosis. So that means all the steps involved in meiosis that leads to genetic variation in gametes will not happen here. This is a very important understanding. Okay? So it is only inherited maternally. So all the family members from the maternal lineage will have inherit the same mitochondrial DNA. So easier, some of you say easy to trace the lineage, easy to trace a common ancestor because they are always transferred from egg to egg, okay, to the maternal family. So finally, now this one, so we already mentioned that the alleles will not change in the DNA when it's passed to the offspring by binary fission. But one thing that will cause alleles to change a lot is the mutation. Mutation rate is higher in mitochondrial DNA because mitochondrial DNA is naked. Naked means there's no histone protein to protect the DNA from mutation, so there's a higher rate of mutation. And also we want to learn there is no enzyme to repair the mutation. Okay, like for eukaryotes, when they carry out DNA replication, they use DNA polymerase, right? Polymerase. Okay, so they use DNA polymerase to extend the DNA strand by adding the nucleotide, right? So after the new DNA strand is formed, they also use another DNA polymerase to check the mistake. That means any base that's added wrongly, any nucleotide added wrongly, and correct it. So they remove it and replace with the correct nucleotide. So this the eukaryotes can do, but the bacteria cannot do. Okay, so therefore, once mutated, it'll be mutated, so the rate of mutation will be higher. Okay, faster molecular clock. Faster molecular clock means what? It means that the bacteria multiply faster than eukaryotes. So average time one bacteria multiply is every 20 minutes. every 20 minutes, the cell will replicate, the cell will divide. So therefore, they will produce, replicate, means the DNA will replicate every 20 minutes. So the more times it replicates, the more the chances of mutation. So that's what it's trying to say, faster molecular clock. Okay? Now, some students say, oh, the mitochondrial DNA can mutate at a constant rate. No, mutation is never constant. Mutation is always by chance. Like a very good illustration is you eat your lunch and then you chew, suddenly you bite your tongue. So this is by chance. You do not know when you bite your tongue. Okay? So mutation is always by chance, it's not constant one. Right? So number 10, so more choice of suitable sections of mitochondria to test, more accurate time estimate. So because mutation rate is quite high, so we can choose a region of the mitochondria where it's subjected to mutation, more mutation, and then use that as a biological clock, right? The more mutation, the more differences means it mutate more time, means it already divided more time. So the more differences they are, okay? Why division can cause mutation? Every time the cell divide, DNA has to replicate. When DNA replicate, wrong nucleotide can be added in, wrong base can be added in. So therefore, replication, let me write in words. When the cell divides, then the DNA must replicate. Then there could be mistakes. Mistake in terms of wrong nucleotide or base added, so lead to change in the base sequence of the DNA. So therefore, that means the higher the rate of cell division, the higher the DNA replicates, rate of replication, the higher the mistake, the higher the change in base sequence, the higher the mutation rate. Okay? Okay, next question. 4B. Today I go into the topic quite detailed so that I hope you can learn from there. So I go into quite detail. Tei Shun Sin is outside. Okay, let's admit. Okay, Tei Shun Sin. Why is this? Okay. So the scientists studied the effect of increasing the number of tourists each year in the number of cases. Let's look at the graph. So the graph is showing what? Number of tourists visiting the Sunda Islands each year in 1,000. That means every value here you must times 1,000, okay, to get the total number of individual. Y-axis is the number of recorded invasive species. So you can see that there is a straight line going up. When the number of tourists increase, the number of invasive alien species also increase. Okay, hang on. I need to start share again because something was hanging just now. This one. Okay. Okay. Right. So calculation has been done for you. You only need to do the analysis here. So Pearson linear correlation has been carried out, calculation. And the scientists got the R value is 0.30. Okay, so the scientists concluded that there is a significant correlation between the number of tourists and number of invasive alien species. So table 4.1 shows the table of critical values for Pearson linear correlation. So with reference to table 4.1, explain why the R value of 0.930 indicates that there is a significant correlation between the number of tourists and the number of invasive alien species. So this is question number 4. Okay, so the mark scheme is 2 marks. So first mark is given for, okay, I show you the arrow, then you know how the examiner mark, look for the answer, okay? First, you must get this correct, and next, you must get this correct. So that means you know which one is the correct one to refer to make conclusion. So how many total number of observations there are in the graph? So please check first. So in the graph, how many crosses there are? 1, 2, 3, 4, 5. 5 crosses means 5 data. Okay, 5 data. Okay, 5 data. So therefore, the number of observation is 5. So you have to choose this one here. Okay, under number of observation is 5. Then your calculated value is 0.93 is here. 0.93 is here. Okay, so therefore, you also need to know that you are concluding based on this figure here, 0.05. Okay, so how do you write your answer? You say that for n equals to 5, you can say that you can use critical R value is 0.878. Okay, 0.878. So, and then the calculated R value 0.930 is 0.878. So then you get the full marks. Okay, so either you refer to the critical R value, which is 0.878, or you refer to 0.05. So another marking point is at n equals 0.05. So the probability, okay, is between 0.01 to 0.05. Therefore, probability is less than 0.05. Probability is less than 0.05. Okay? For n equals to 5. So that will give you 2 marks. So that means first mark is 5 is the number. Second mark is either 0.05 or 0.875. So you need to conclude, the R value is more than 0.878 or the probability is less than 0.05. Okay? So these are the 2 marks given. Any question? Okay, next. Part 2, the introduction of alien species is one of the reasons that leads to the extinction of Bali and Javan tigers. Suggest 2 other reasons why these 2 subspecies become extinct. So this one is very straightforward, right? So other than introduction of alien species, what else can lead to the extinction of the species? This one is theory. So directly apply from theory. Okay. So we have habitat loss. More is a keyword. So if you write competition, there's no mark because competition exists naturally, right? Competition exists naturally. Why can last time got competition, no extinction, now got competition, got extinction? You must say there are more. The keyword is more. A lot of students didn't write this. That's why you write competition, no mark. So there are more interspecific, intraspecific competition for what? So you need to spell out clearly, crystal clearly to the point, right? This is animal, this tiger. They compete for what? They compete for mate, they compete for space, right? They compete for prey, yeah, right? Prey, okay? And then what else? Because of poaching, hunting, poison by human because of disease. Now, some students write climate change. I will not give mark for climate change because climate change affect the plant more than animal because plant cannot move, but animal can migrate. So if it's too hot, when there's no water, or when it's too cold, they can always migrate, but plant cannot migrate. So plant affected by climate change. The mark scheme doesn't give mark for animal, climate change affect animal. Okay, so you must give better answer than just climate change like that. There are a few more better answer than this, right? So you must choose the better answer to write in exam to secure the mark. Okay? Don't just, !aiya! Chin-cai lah, just write. Cannot chin-cai one, must write the correct one. I'm having problem here. Okay. Right, 4C. 4C, suggest why PT Sumatrae is still, one thing, so I noticed still a lot of students, about one-third or one-quarter of students did not underline when they write species name. Species name must underline. You already saw it's italic, so must underline. Please, please, please. Biology student, right? Okay, you know the format of writing. Okay, right. So why PT Sumatrae is still considered to be a member of the species Panthera tigris? That means some students, they don't answer this question. They actually ask, this is the kingdom, the kingdom, that chapter, the question from there. Why PT Sumatrae is still considered to be a member of the species Panthera tiger? That means, why are they considered as the same species? So, two individuals considered the same species because definition, they are morphological or physiological or behavioral or biochemically similar to the Panthera tigris. So why this subspecies is considered same species as Panthera tigris? Because they are similar to them. Okay. So first mark. Second marking point is they are genetically similar to. Genetically similar to means they may have similar, keyword is similar, not the same. If you write same, no mark. Similar means roughly the same genes. They will have the same genes. Sorry, similar genes, similar alleles, similar base sequence in DNA, similar karyotype, similar DNA, similar chromosome number, similar mitochondrial DNA. Okay? Right? So first one is morphology, physiology, behavioral, biochemical. Second one is genetic. Third one is because they can interbreed with the Panthera tigris to produce vigorous fertile offspring. Okay? So you must spell out fully, well, you must explain fully. Some students say, they can reproduce. I reject reproduction because you must describe how to reproduce, able to interbreed with the Panthera tigris to produce vigorous fertile offspring. Not they can reproduce. Okay, they must reproduce, so what? The offspring must be fertile and vigorous. Okay, right. So let's go to the next question, 5A. 5A. So what is this? Mitochondrion, right? Very interestingly, they tell the story. So they have got this mitochondrion. So I'll draw the mitochondrion. Yeah, square. I don't want square. Mitochondrion. So I'll draw the mitochondrion. Okay. So we have a mitochondrion here. And inner membrane is highly folded. So let's draw the inner membrane, highly folded. Okay. Very, very. I draw a few will do, just to illustrate. Okay. Right. Dot, 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 dot. Okay. So intact mitochondria was removed from the cell. So the cell contain a lot of mitochondrion. So those mitochondrion, okay, are removed from the cell. So this is one mitochondrion removed from the cell. Okay. And then a technique is used to remove the outer mitochondrial membrane. So they use a technique to just remove this outer membrane only. Okay. This was being removed, so no more there. Okay. And then they leave the inner membrane is intact. So inner membrane is not removed, it's intact. Then the inner mitochondrial membrane was separated from the contents of the matrix so that both could be analyzed. So they separate the inner membrane and also they separate the matrix, and they want to analyze these two components separately. The removal of the outer membranes of mitochondria involves placing the organelles in pure water. So how do they remove the outer membrane? They put the mitochondrion into water. So they put this whole thing into water. Okay? Pure water. Means psi equals to 0. Correct? Yes. Psi equals to 0. psi s equals to 0. This results in the rupture, bursting of the outer membrane only. So the inner membrane does not rupture, which is very interesting, right, and remains intact. So question asks the student, suggest and explain why the inner membrane mitochondrion remains intact when the organelle is placed in pure water. So the answer is very interesting one. So firstly, you see, the inner membrane is not freely permeable. Okay, let's look at the mark scheme. Okay. So very interesting. Now, as the water enters the mitochondrion, the outer membrane expand and break. Okay, but the inner membrane of mitochondrion is highly folded. So when it's highly folded, okay, when they expand, right, they can actually expand a lot and they will not break because they are highly folded, so they can expand. So first marking point. Second marking point, when water enters the matrix by osmosis down water potential gradient, the okay, point number two is related to point number one. Okay, so when water enters the mitochondrion, outer membrane will burst, inner membrane will stretch. So inner membrane does not break. Okay, so first two are related to each other. Then next one is the inner membrane is impermeable to water. So this is another point already. Impermeable to water. Okay, so folded is first marking point. Impermeable is a property, it's the second marking point. So because of that, water can enter. So this is the outer membrane, right? So I draw outer membrane here. Okay, suddenly. So water can enter here. Water can enter, water enters here. But water cannot enter here. Water cannot enter. So it's impermeable. So therefore, water enter, then the membrane stretch and burst, outer membrane. Inner membrane, the water cannot enter, so it will not change shape. Okay, it will not stretch the inner membrane. So water does not enter the matrix by osmosis down water potential gradient. Okay, so these two are related. Lastly, the inner membrane can pump proton out of the matrix. So this is the inner membrane, right? Is able to pump the proton into the intermembrane space when electron is transported in the mitochondrion. Okay, so let's go to the hmm, respiration topic. We're going to look at mitochondrion, inner membrane of mitochondrion. What does it contain? So we have to look at oxidative phosphorylation. Okay, this one? Here, this one. Okay, here, right? Yeah, yeah. So this is inner membrane of mitochondrion. This is inner membrane of mitochondrion. This is outer membrane of mitochondrion. This is intermembrane space. So you see that when electron is passed along the ETC, right? Proton is being pumped, okay, from the matrix to the intermembrane space. So because proton is being pumped out, and this proton will reduce the water potential here. Okay, you reduce the water potential in the intermembrane space, and that will actually cause water to come out from the matrix, so the inner membrane will not burst. Okay, so hence the mark scheme answer here says that inner membrane moves the ions out of the matrix so that less water can enter by osmosis down water potential gradient. So it's actually decreasing the gradient of the for water to enter, right? So initially is very low. So this is matrix. So This is the intermembrane space. This is the matrix. Okay? So I need to write it. This matrix, and this is the intermembrane space. Intermembrane space. So initially it's low. When the proton is being pumped out, then you know that the water potential inside the matrix will actually, sorry, what's being pumped out? Proton, right? So less substrate, so water potential will increase. Less, not substrate, less solutes. So the gradient, water potential gradient will become less steep. Okay, less steep. So water movement into the matrix becomes slower. The rate becomes slower. So it will not burst. Okay? Right. Last question. Name 3 molecules other than coenzymes that are found in the mitochondrial matrix and explain their role in aerobic respiration. So this one is very easy. But some students, they didn't score the marks because the question already said don't mention coenzyme. So what are the coenzymes here? NAD plus, FAD plus, and also the acetyl. CoA. Okay, the CoA. So these 3 cannot mention because they said other than coenzymes. So other than that, any other molecules involved in the aerobic respiration in the matrix will have marks. So I have pulled out just now this oxidative phosphorylation. Okay. Here. Okay. So this is matrix, right? So what can you find in matrix? Pyruvate. Okay. OAA, oxaloacetate, citrate, alpha ketoglutarate. So these are the components we can find in the matrix. So let's look at the mark schema. 3 molecules. So state the name of the 3 molecules and explain their role. So what do they do in aerobic respiration? So let's look at the mark scheme. Okay. Number 1, pyruvate. Pyruvate is used for what? It's used in link reaction. Okay. It's supposed to combine with CoA to form acetyl-CoA. So practically you can also write the chemical equation. Okay, pyruvate plus CoA to form acetyl-CoA. Correct? Correct. Pyruvate plus CoA becomes, actually it's not correct. Pyruvate becomes acetyl, okay, correct. Pyruvate plus CoA becomes acetyl-CoA. Correct. The balance equation is correct. Okay. Then we have oxaloacetate. What does it do? It will accept the acetyl to form citrate. So write the full chemical equation. Then the citrate will be dehydrogenated to form alpha ketoglutarate. Okay. Then you can also name the enzymes that involve like dehydrogenase, decarboxylase, okay, that are used in link reaction and also in Krebs cycle. Then oxygen is involved. Yes, oxygen is involved in the matrix, right? It is in the matrix near to the inner membrane that it will accept the proton and electron to form water. So you must say that oxygen accepts electron proton to form water. So what does it do? Okay, water acts as a solvent for all the substrate involved from number 1 until number 5. And then, of course, what other molecules? DNA, RNA are molecules, right? They are nucleic acids, they're molecule. Ribosome is also a molecule. Ribosome is a nucleoprotein, right? RNA plus protein, so it's nucleoprotein. So they're involved in the synthesis of protein used in the mitochondrion, like enzymes, dehydrogenase, decarboxylase, carrier proteins, electron carriers, okay, stop particles, and so on, any named protein, and one other correct marking point. Okay, so I will stop here today and continue tomorrow. Right, stop record.